Antiviral Properties of Sunlight, Part 4, Defense in Depth. In this YouTube, we discuss how 3 innate immune hormones provide a layered defense system against viral infections. These hormones are connected to the impact of solar radiation on the human body. These hormones are calcitriol, 125 vitamin D3, calcifediol, 25 vitamin D3, and melatonin. All of these hormones share a common action. They all upregulate the master inflammatory antioxidant transcription factor, NRF2. NRF2, in turn, upregulates a key antioxidant enzyme, GPX4, glutathione peroxidase 4. Glutathione peroxidase 4 chemically reduces reactive oxygen species as well as peroxidated lipids, which are upstream drivers of the programmed cell death pathway known as ferroptosis. It is now recognized that ferroptosis is often a key player in viral infections. In this YouTube series, we have been discussing sequential viral transactivations, such as the reactivation of herpes viruses in response to COVID-2 infections. In this particular YouTube, we will now discuss how organelle injuries can set up conditions of oxidative stress which culminate in ferroptosis, and ferroptosis may be a key driver of sequential viral transactivations. For that reason, utilizing these 3 innate immune hormones as a means of suppressing ferroptosis may be a potential way of suppressing sequential viral transactivations, for instance, in the context of COVID and long COVID. To organize the discussion of defense in depth, it is useful to begin with some analogies. For instance, the defense in depth strategies of trench warfare, first developed in World War I. Defense in depth in trench warfare is a strategy using multiple layered and interconnected trench lines rather than a single front line. Developed during World War I to counter superior firepower, this approach slows advances, drains attacker resources, and utilizes obstacles like wire and mines to prevent breakthroughs, allowing for counterattacks from reserve lines. The concept of defense in depth is also used in computer cybersecurity. Defense in depth, layered defense model. Defense in depth is a layered cybersecurity strategy using multiple redundant controls, physical, technical, and administrative, to protect assets, ensuring that if one defense fails, others contain the threat. Key layers include security policies, perimeter defenses, i.e. firewalls, network security, endpoint protection, application security, data encryption, and crucially, user training. These elements of cybersecurity can be analogized to functions of the innate immune system as well as the adaptive immune system. Here we are concentrating on one specific aspect of the innate immune defenses, namely the redundancy of these 3 hormones, melatonin, calcifediol, and calcitriol, their ability to suppress ferroptosis, and in doing so protect tissues and slow or impede viral infections. Now, these concepts connect to this recent review: layers of defense, protection from respiratory viruses by epithelial intrinsic immunity, innate immunity. A central challenge in defending mucosal barriers is protecting against pathogens while also limiting excessive inflammation. Respiratory viruses are a prime example. Respiratory viruses present a threat to their target cells, the epithelial cells that line the airways, but excessive leukocyte recruitment to fight the infection can lead to inflammation and respiratory distress. This review focuses on how epithelial intrinsic defenses contribute to achieving a balanced antiviral response by adding layers of defense that engage in sequence to control infections. These layers include secreting a defensive extracellular barrier, directly blocking viral replication through cell-intrinsic effector mechanisms, amplifying cell-intrinsic defenses within the epithelium through type 3 interferons and other epithelial-specific mechanisms, and 4, coordinating leukocyte recruitment and activation. Recent findings in humans and organoid tissue models support the idea that the layers of defense created by epithelial intrinsic mechanisms frequently and successfully counteract respiratory virus infections and limit their health impact. A sidebar comment. The ability of innate immune hormones such as melatonin, calcifediol, and calcitriol to boost antioxidant defenses and thus suppress ferroptosis is a key way that viral infections can be impeded not only within epithelial sheets in mucosal tissues, but within deeper tissue layers, the parenchyma of organs. In addition, circulating immune cells or immune cells that have been recruited to a site of infection need to be protected from microbial-induced ferroptosis. And this is one of the key roles of these innate immune hormones. And what we see here in these information vectors is a redundancy, a very important redundancy that gives defense in depth to this key innate immune defense, namely to suppress ferroptosis. And this is a figure from the review. Layers of defense model in which sequential engagement of mucosal defenses control the infection while minimizing unnecessary inflammation. Again, the ability of these immune hormones to upregulate antioxidant defenses fits into this model. The first 3 portions of this series can be accessed from these URLs. Here we are emphasizing the concept of defense in depth, which is derived from hormone action redundancy, and in this case, the ability of these hormones to boost NRF2 and GPX4, and by doing this, suppress the process of ferroptosis. Later, in the 5th part of this YouTube series, we will concentrate on how viruses have developed countermeasures that impair these innate immune functions to suppress oxidative stress and ferroptosis. A sidebar comment. When you have defensive systems that impede a pathologic process, when you inhibit the defenses, you activate the pathogenic process. In this case, viral countermeasures by inhibiting melatonin, calcifediol, and/or calcitriol will be promoting ferroptosis. And that is part of the strategy of propagation in a number of these pathogens. In the second part of this YouTube, part 4 of this YouTube series, I would like to draw on this graphical summary of concepts, illustrating sequential viral transactivations. Steps 1 through 4, beginning with a primary infection such as CoV-2. The CoV-2 infection can frequently upregulate dormant herpes viruses such as herpes simplex virus 1 HSV1, or Epstein-Barr virus, EBV. These herpes viruses, in turn, can upregulate dormant fossil viruses known as human endogenous retroviruses, HERV viruses. This is a transactivation of these dormant ancient fossil viruses in the human genome. Many of them encode proteins, and these proteins can be very toxic. For instance, HERV proteins are central to the pathogenesis of multiple sclerosis, ALS, and, we'll see, schizophrenia. In sum, this set of sequential viral transactivations creates a polymicrobial pathogenesis. It is important to note that the upstream drivers of this polymicrobial pathogenesis need not be a primary viral infection. In fact, a single toxin from a virus such as CoV-2 could potentially reactivate dormant herpes viruses and set off this chain of events leading to sequential viral transactivation and polymicrobial pathogenesis. New literature is appearing on PubMed that reinforces this concept. HERV-W, this is a retrovirus in the human genome, its envelope protein, abbreviated ENV. So HERV-W envelope protein transcription in B cells, predicting symptomatic COVID-19 and risk for long COVID, can express a full-length protein despite a stop codon in messenger RNA from chromosome X via a ribosome read-through. This paper, published in 2025, is particularly important. It provides key molecular genetic information about the transactivation of a HERV virus and its toxic protein. Once again, this transactivation of the HERV-W envelope protein is occurring both in acute COVID as well as long COVID patients. Reading from the abstract, I am going to modify a couple of sentences for clarity. The human genome contains 8% endogenous retroviruses, HERVs. Though HERVs contribute to physiological functions, copies of HERVs retain pathogenic potential. The HERV-W envelope protein was shown expressed in patients with worse COVID-19 symptoms and post-COVID syndrome, PASC. A significant detection of the messenger RNA encoding HERV-W envelope protein from patients with COVID-19 in B cells from RNA sequence reads obtained from peripheral blood mononuclear cells. These are monocytes. These data stratified with increased COVID-19 symptoms or with post-acute sequelae of COVID, long COVID, after 3 months. The HERV-W envelope U3R RNA was confirmed to display the best alignment with chromosome X. ERVWE2 locus, that is a locus on the X chromosome. Moving to the end of the abstract. Results not only confirm HERV-W envelope RNA origin in these patients, COVID and long COVID patients, but show for the first time how a defective HERV copy can be translated into a complete protein when specific factors make it possible at the ribosome level. The present proof of concept now requires further studies to identify the factors involved in this newly understood mechanism following SARS-CoV-2 exposure. And this is a portion of a figure from this paper. It shows the levels of HERV-W envelope protein, a portion of the retrovirus's RNA. That is what U3R represents. These are the RNA codes that were read from patients with post-infection groups 1 and 3, later diagnosed as either having PASC or no PASC. In other words, these patients had COVID infections, and group 1 developed PASC, post-acute sequelae of COVID, long COVID symptoms. They're looking at blood monocytes, and what they find is that there's elevated levels of the HERV-W envelope protein, RNA, in the people who have PASC. And these results confirm and extend an earlier report from 2022, persistent HERV expression in patients with post-COVID symptoms. There is a large effort to develop small molecule inhibitors of HERV proteins. And here's an example of an inhibitor being developed to block the functions, the toxicity of a HERV-K protein. Now, I mentioned earlier that viruses have developed attack strategies to cripple the defenses, the antioxidant defenses of cells and tissues. And we discussed how HERV viruses are frequently drivers of neurological disorders. This paper is from 2024. Ancient dormant virus remnant HERV-W1 drives ferroptosis via degradation of glutathione peroxidase 4 and the heavy side chain of the cysteine glutamate antiporter, SLC3A2, in schizophrenia. And so these schizophrenia patients have a double loss in antioxidant capacity, and this is driving ferroptosis. Let me have a sidebar explanation of that. GPX4 is a key antioxidant enzyme which utilizes glutathione to chemically reduce reactive oxygen species as well as peroxidated lipids, which are the upstream drivers of ferroptosis. The cysteine glutamate antiporter is critical for the synthesis of glutathione, the substrate molecule for GPX4, glutathione peroxidase 4. You need glutathione and the enzyme to process glutathione, use its energy to chemically reduce the reactive oxygen species and the peroxidated lipids. So if you lose both the enzyme and the antiporter in the cell membrane needed to pull in cysteine, an amino acid which is the precursor to cysteine, which is used to build the tripeptide glutathione, this is very damaging and will promote ferroptosis in cells. If you utilize MRI to examine the brains of people with schizophrenia, this is a paper from 2005, Neuroimaging in Schizophrenia. What does it tell the clinician? These are T2 weighted MRIs of monozygotic twins, one with schizophrenia compared to his twin. The twin who has schizophrenia has very enlarged ventricles. So this is a macroscopic manifestation of the pathophysiology underlying this neurological disorder. And what's remarkable is that it has now been connected in part to the reactivation of this ancient dormant virus, HERV-W1. In parts 1 through 3 of a prior YouTube series on this channel, we discussed viral transactivations during COVID. In part 3 of this series, we discussed neuropathologies that emanate from these viral transactivations. The URLs to these YouTubes are highlighted in blue. Now I would like to move to the topic of therapeutics for the treatment of viral transactivations. And these therapeutics can potentially be used to suppress the viral transactivations from occurring. In the upper left of the slide, you'll see a set of therapeutics delimited with curly brackets. These are the mathematical symbols for a set. This is a therapeutic toolbox which includes melatonin as well as vitamin D receptor agonists. Vitamin D receptor agonists include the hormones calcifediol, 25-vitamin D3, which is the metabolite that health professionals commonly use to assess, quote, vitamin D status in the serum. That is an unfortunate misnomer because what is being measured is 25-hydroxyvitamin D, the liver metabolite, not the skin metabolite, inactive vitamin D3. This causes constant confusion between patients, practitioners, even in scientific literature. So it's very important to define the state of hydroxylation of these secosteroid hormones. So calcifediol is 25-hydroxyvitamin D3. It can be abbreviated several ways, 25-vitamin D3 or 25D3 or simply 25D. This again is the, quote, liver metabolite in the serum. It's also secreted in exocrine fluids such as tear fluids and bile. Calcitriol is the, quote, kidney metabolite. It is doubly hydroxylated. It is 1,25-dihydroxyvitamin D3, abbreviated 1,25-vitamin D3 or 1,25D3 or simply 1,25D. All of these terms are found in scientific and clinical literature. Finally, a very important synthetic analog of calcitriol, paracalcitol, which is the preferred therapeutic used to treat chronic kidney disease patients. Paracalcitol has been recently repurposed for the treatment of herpes simplex virus keratitis, which we will describe shortly. Paracalcitol is not based on a vitamin D3 backbone. It's based on a vitamin D2 backbone. That's the secosteroid backbone. It too is hydroxylated at the 1 and 25 carbon positions, similar to calcitriol. So it can be abbreviated 1,25-vitamin D2 or 1,25D2 or simply 1,25D. Again, all of these abbreviations will appear in different portions of the scientific and clinical literature. Here are two papers that describe the importance of vitamin D receptor agonists for the suppression of herpes simplex virus 1. The association between serum 25-hydroxyvitamin D, this is calcifediol levels, and the prevalence of herpes simplex virus. These are human studies, and there's an inverse correlation. The higher the serum vitamin D level, again this is 25-hydroxy D levels, serum calcifediol levels, the lower the incidence of herpes simplex virus in the body. The second paper, 25-hydroxyvitamin D3, again this is calcifediol, and 1,25-dihydroxyvitamin D3, calcitriol, as an antiviral and immune modulator against herpes simplex virus 1 infection in HeLa cells. these two papers set the stage for a discussion of this paper and others. Herpes simplex virus 1 in the brain, the dark side of a sneaky infection. This paper describes how HSV1 can reactivate from neurons within the trigeminal ganglion, spread anterior grade out to facial dermatomes, creating cold sores and other ulcerations. In addition, the infection can spread from the trigeminal ganglion to the brainstem and beyond, creating an encephalitis, a viral encephalitis. And it has now been established that HSV1 can move through these anatomical tracts into the brain and produce disorders such as Alzheimer's disease. In addition to creating cold sores in the orofacial area, HSV1 can move through the ophthalmic branch of the trigeminal nerve into the eye. And from there, the virions can spread to the cornea and infect the cornea. And at this point, it is called HSV1 keratitis. It's a viral infection of the corneal epithelium and stroma. So, therapeutics for viral transactivations. Herpes viruses such as HSV1 can be transactivated in response to other viral infections, such as a COVID infection. So these two papers describe the successful use of vitamin D receptor agonists for the treatment of ongoing HSV1 keratitis and provide additional evidence that continued use of these VDR agonists can be used to suppress future reactivations of the HSV1 keratitis. Calcifediol and paracalcitol as adjunctive therapies for HSV1 keratitis and corneal perforation, a case report. Second paper, herpes simplex keratitis and vitamin D receptor agonist, 2 case reports. The vitamin D receptor agonist used here was calcitriol. So 3 different vitamin D receptor agonists being used to treat HSV1 keratitis. Calcifediol, 25 vitamin D3, paracalcitol, 125 vitamin D2, and calcitriol, 125 vitamin D3. All of these VDR agonists would be expected to upregulate the master anti-inflammatory antioxidant transcription factor, NRF2, which in turn upregulates GPX4, glutathione peroxidase 4, which is a key enzyme to reduce reactive oxygen species and peroxidated lipids and suppress ferroptosis. But here, through steps yet to be determined, the vitamin D receptor agonists, by stimulating these pathways, it is now known that boosting NRF2 and or GPX4 is able to suppress HSV1 infections. Why is this of general interest? Herpes viruses may contribute to Alzheimer's disease via transposable elements. HSV1 is associated with Alzheimer's-associated transposable elements in infected Alzheimer's-affected brains, which may be reversed through available drugs. Sidebar comment. Some of these available drugs are vitamin D receptor agonists, calcifediol, paracalcitol, and calcitriol. In addition, melatonin may be of particular use because there are some reports that melatonin can stimulate vitamin D receptors, potentially activating these pathways. So summarizing these into a sort of a graphical summary, the adjunctive therapies which we've been discussing could be potentially used with existing pathogen-directed therapeutics like acyclovir. But here we're talking about host-directed therapeutics to bolster innate immune responses, innate immune defenses. So stimulating a vitamin D receptor, VDR, with a natural agonist such as calcitriol, the kidney metabolite of vitamin D3, 125 vitamin D3, serves to suppress HSV1. And we see that other vitamin D receptor agonists can also achieve this. Calcifediol and paracalcitol can be used together to suppress an ongoing infection. And in this case, the paracalcitol was discontinued after the acute infection was over and the patient was maintained for over a year using calcifediol alone. So summarizing all of this into a concise information vector, using a set of vitamin D receptor agonists, and this is an and/or set. You can use combinations of these. It's a toolbox. Calcifediol, paracalcitol, or calcitriol to stimulate vitamin D receptors, upregulating NRF2 and GPX4 to suppress HSV1, to suppress viral keratitis. Now, elsewhere on this channel, we have discussed various COVID toxins, and one of the virulence factors of CoV-2 is the CoV-2 envelope protein, also known as the E protein. In the bottom right of the slide, you'll see a link to a URL. This is from March 2025. This is a update on the CoV-2 envelope proteins, its cytotoxicities. Currently, there are over 10 cytotoxicities known to be associated with the envelope protein. It is a major virulence factor for CoV-2 infections and likely to be contributing to the pathogenesis in long COVID. Here is a recent review that I highly recommend, a comprehensive review of current insights into the virulence factors of SARS-CoV-2. In the case of the envelope protein, you'll see here in this figure, the right side of it, in small letters, envelope protein. One of the key pathologies associated with the envelope protein is a viral porin. A porin is a molecule that forms a pore in a cell membrane. Viral porin is the term used for viral specific porins. In the case of the envelope protein, it can porate not only the cell membrane, but also organelle membranes, such as lysosomes and the Golgi apparatus, collapsing key proton gradients needed for their function. And by discharging the acidic compartment within lysosomes, this allows the virus, virions, to accumulate within lysosomes and then be discharged when the lysosome fuses with the cell membrane. That was an early recognized means of viral propagation in the early days of the pandemic. In addition, this envelope protein can insert into mitochondrial membranes, discharge proton gradients there as well. So organelle injuries are frequently occurring in viral infections. This is part of their strategy to hijack organelle functions, in some cases destroy them. Peroxisomes, for instance, are degraded in COVID infections. So as cells and tissues are weakened by a COVID infection, in these environments, this is where viral transactivations can occur. For that reason, it's very important to emphasize the potential utility of host-directed therapeutics, such as melatonin and vitamin D receptor agonists, to bolster host defenses against potential viral transactivations. In addition to the herpes viruses, there are the threats of HERV virus transactivations, and these too may be protected against by the use of melatonin and vitamin D receptor agonists. So summarizing this in a graphical summary, combining host-directed therapeutics such as sunlight, near-infrared light from LED sources to stimulate melatonin, combining those with pathogen-directed therapeutics such as acyclovir, the standard of care for HSV-1 infections, you arrive at adjunctive therapy strategies. And that was actually used in the paper which discussed earlier, where the individual who had HSV-1 keratitis and corneal perforation, she was first treated with acyclovir, discharged from the hospital, but the HSV-1 infection reappeared, and at that point they utilized the host-directed therapeutics, calcifidiol and paracalcitol, in combination with acyclovir to suppress the recurring HSV-1 infection. A sidebar comment. In the green information vector underneath adjunctive therapy strategies is a key concept, a takeaway concept. Melatonin and vitamin D receptor agonists can be potentially used for long periods of time. They are Nrf2 boosters, and that is a common strategy that needs to be emphasized to boost antioxidant capacity of cells, to achieve defense in depth in this critical immune dimension. For long-term treatment of IAC conditions, infection-associated chronic conditions, patients will be treated with pathogen-directed therapeutics episodically. However, for long-term bolstering of the immune system, host-directed therapeutics are needed. And that's where this toolbox, melatonin, vitamin D receptor agonists, are a potential way of achieving that. In part 5 of this series, we are going to talk about how pathogens attack these critical transcription factors and enzymes that are needed to fight pathogen-induced ferroptosis. We introduced this slide in a previous YouTube, part 3 of this series, and a link to that YouTube is highlighted in blue at the top of this slide. This paper, Critical Role of Ferroptosis in Viral Infection and Host Responses, contains this wonderful diagram. It is a metabolic map of viral ferroptosis. This is a compilation of decades of study, billions of dollars of research. So the attributes of this diagram is that it summarizes key attack strategies that different viruses use to disable the antioxidant defenses, the depth and defense system that cells use to prevent ferroptosis. Some of the viruses attack Nrf2 expression. Other viruses attack GPX4, either its expression or to promote its degradation. And as we have seen in the case of schizophrenia, the reactivation, the transactivation of a dormant fossil HERV virus, its toxic protein degrades both GPX4 and a component of the cysteine glutamate antiporter, which is abbreviated XCT. So all of these are, quote, Achilles heels of cells. If viruses are able to cripple any one of these critical nodes in the antioxidant defense in depth system, this makes the cell prone to the accumulation of reactive oxygen species as well as peroxidated lipids, driving the cell towards programmed cell death known as ferroptosis. This concludes our discussion of part 4 of this series. We will proceed to part 5 of this YouTube series on antiviral properties of sunlight, dealing with the issue of these viral countermeasures to the oxidative defenses that cells utilize to combat viral ferroptosis. In closing, I would also like to draw your attention to several YouTubes recently posted on this channel, which deal with ferroptosis being driven by GPX4 silencing, and this occurs by GPX4 promoter methylation. This type of silencing of the GPX4 gene can occur from pathogens, toxins, but also by ischemic reperfusion injury. And ischemic reperfusion injury is common in COVID and long COVID patients because of microclots. There's also a very good reference here at the bottom of the page, GPX4 in cell death, autophagy, and disease. If you would like a PDF slide summary of this particular YouTube, which contains active links to other YouTubes on this channel, please go to the Google Docs folder highlighted in green at the bottom of the slide. Here you will see a set of subfolders. If you'll navigate to the subfolder Download First, you'll be able to locate the PDF slide summary for this particular talk more easily. There's also subfolders on this Google Docs folder pertaining to ferroptosis and COVID, ferroptosis and ferritinophagy, as well as GPX4 gene silencing. You're welcome to download all the material on this Google Docs folder. With that, we'll pause briefly and then proceed to part 5 of this series.